Hello everyone. Today we are going to break down the structure and function of antibody in a way you will never forget. Let's start. Now your antibody is like a Y-shaped defender. Okay? It's like a Y-shaped sword. It's sharp and it's precise. Now let's see the definition of your antibody. Antibody is also known as immunoglobulin. So in case you see this term, don't get confused. Clear? Antibody or immunoglobulin is a specialized glycoprotein. It's protein in nature produced from activated B cell, that is plasma cell, in response to an antigen, and it is capable of combining with it. Okay. So if this is the activated B cell, okay, activated B cell producing what? Antibody. Producing your antibody in response to what? In response to this antigen, okay, this antigen. Then the antibody which is produced here is capable of combining with this, okay, that is it can combine with this antigen. This is the definition, okay. Now let's move ahead. Now your antibody is Y-shaped, okay. It's Y-shaped and it is made up of 4 chains. What are these 4 chains? You see, we'll see the first 2 chains. So you see this chain, okay. These 2 chains, you can see these 2 chains are muscular, they are strong, they are heavy. These two are the heavy chain. They are like strong arms, okay. So your 4 chains has first 2 heavy chain, followed by, you see next to heavy chain, this chain. These 2 chain, what are they? They are not as strong as your heavy chain. They are lighter in comparison. They are like sidekick to the strong arm. They are light chains. So these are 2 light chains. So in your 4 chains, you have 2 heavy chains and 2 light chain. Now both these heavy chains are identical to each other. Okay, remember this. And both these light chains also will be identical to each other. Clear? Now you see, what are these 4 chains held together by? Can you see this? This iron bond, okay, this strong bond between them. What is this? This is the disulfide bond, okay. This bond is the this bond, just holding the 4 chain together is the disulfide bond, okay, clasping it together. Now apart from the disulfide bond, we have also certain weak forces like your hydrogen bond, your non-covalent interaction. All of them are doing what? They make you one strong defender, one strong antibody to protect us, हमें protect करने के लिए against antigen. Clear? Now apart from these, each chain also of your heavy and light chain ends at the upper end as the amino terminal, okay. This is the amino terminal, NH3. Both heavy and light. Both heavy and light will have NH3. And the lower end is called carboxyl terminal, okay. That is COOH, okay, COOH. Now for the light chain, this is the upper end and this is the lower end. So this will be COOH and this will also be COOH. Understood? Upper is the amino terminal and lower is the carboxyl terminal. Clear? Now let's see each of these chain individually. Now you see the heavy chain. The heavy chain, there are 5 classes of heavy chain. Each of these 5 class gives your antibody an identity. The classes of antibody, 5 classes of antibody are based on these heavy chain. Okay, you see what are these 5 heavy chains? These are gamma, which is the IgG antibody, your alpha, which is present in IgA antibody, mu present in IgM, epsilon present in IgE, and delta present in IgT. Clear? This is the heavy chain making your classes of antibody, naming your classes of antibody. Clear? Now let's see the light chain. Light chain is simpler. It doesn't have 5 classes. It has only 2 classes. This light chain will have only 2 classes, that is your lambda and kappa. Now, in one antibody, only one of them can exist. Either both the light chains will be lambda or both will be kappa. Never both together doesn't exist. Okay, it's not present like that. Clear? Now, let's see the class of antibody, what heavy chain it will have and the light chain. Okay, so if it's IgG, G for gamma, the heavy chain will be gamma, light chain will be your kappa or lambda. Now it's IgA, A for alpha, heavy chain is alpha, light chain kappa or lambda. Now it's IgM, heavy chain mu, light chain kappa or lambda. Next IgE, E for epsilon, heavy chain is epsilon, light chain is kappa or lambda. And at last IgD, D for delta, and light chain will be either kappa or lambda. So in the class of antibodies, they can have 5 types of heavy chain, but light chain either will be kappa or lambda. Clear? Let's move ahead. Now, in the heavy chain and the light chain of your antibody, it has 2 zones. Now, what are the 2 zones we will see? First is the variable region, okay, and another zone is the constant region. Now, the variable region, it is the first 110 amino acid residue near the amino terminal, near this terminal, near the NH3 terminal, the amino terminal. The first 110 amino acid residue makes the variable region and the rest makes the constant region. Now, this part is also, this variable region is also called as antigen binding fragment, and this constant region is also called as constant fragment. Okay, now you see why is it called variable region and why is this called constant region? Because it's called variable and constant because in variable region the amino acid sequence is different in different antibodies. In constant region the amino acid sequence is same in different antibodies. Understand it like this. You see this log, okay, all of this log represent different antibodies, okay, different antibodies produced in response to different antigen. These keys represent the antigen. Clear? Now the handle, you see the handle of all log have the same handle even if they are being produced in response to different antigen. Okay, so all these logs have different, all these logs have the same handle. That means this is the constant part, okay, constant part of the log, constant part of the antibody. That is the amino acid sequence here is the same for all the logs. Now let's see the part where the keys goes inside. You see this structure is different. This is the variable region. Understood? This is the variable region, that is amino acid sequence making this part is variable, is different. Clear? Understood? Why it is different? Because the key, the antigen which is going inside has different shape. The antigen making contact with this part of the lock or this part of the antibody is different. So, to fit in, they have different shapes. Clear? Okay. Now, in the variable region, we have certain hotspots. Okay. These hotspots are the part which are more variable, that is hyper variable. Okay, more variable will be hyper variable. So this hyper variable region are the one which actually catches the antigen. Okay, this is known as paratope. The part of antibody which actually makes contact with the antigen, which catch the antigen, is the paratope. How? We'll see this. This is the paratope. This is the antigen. The part of antigen which makes contact with the antibody is called epitope. Okay. So the antibody paratope makes contact with epitope. Okay. Now you see this antibody and this antibody, the paratope structure is different. The variable region structure is different. It is based on the structure of the epitope of the antigen. Understood? Clear? Now, one more point before I move ahead. Now, what did I tell you? The class of antibody is dependent on class of all your antibody is dependent on what? Heavy chain. But after seeing this part of your lecture, what do you think? Which part of heavy chain is responsible for deciding the class of antibody? Is it the variable region or is it the constant region? It is the constant region of heavy chain which decides the class of antibody. Understood? Let's move ahead. Now, let's see. This is the light chain. Okay, this part is the light chain. This is the heavy chain. Clear? This is the variable region, the antigen binding fragment. This is the constant fragment. All these four chains are held together by disulfide bond. Now, let's see the light chain first. In light chain, you have the variable light chain, both side, and the constant light chain. Okay. So, your light chain have how many domain? Your light chain have one variable and one constant domain. Clear? Let's see the heavy chain now. In your heavy chain, this is the heavy chain, okay, variable region of the heavy chain. Now, this is the constant region of heavy chain. Now, you see there are more domains. So, this is the first domain. This is the second domain of your constant region. And this is the third domain of your constant fragment. So, in the heavy chain, there is one variable domain and how many constant domain? 3 constant domain. Understood? Now, you see hinge region. I have never discussed till now anything about hinge region. So, we will see that now. Hinge region is the region which provides flexibility to the antibody. Okay? What is flexibility? That means your antibody can bend to reach its target. Okay? Now, this is made up of PC, proline and cysteine. Okay? It's present in not every immunoglobulin, not all 5 types. It is present only in 3 types: immunoglobulin A, immunoglobulin D, and immunoglobulin G. Okay, immunoglobulin ADG. Clear? It is not present in, the flexibility is not present in immunoglobulin, which one? Me, M and E. So, if it is lacking the hinge region, it should have something in addition. So, your immunoglobulin IgM and E have one variable domain, okay, which is there, but constant domain is 4 constant domain. Okay, so in M and E you have one more domain. This is the constant domain additionally in your IgM and IgE. This is the whole structure of antibody. I hope it was clear. Now let's see the types of antibody. Now the easiest mnemonic to remember the types of antibody is that it's gamed. Okay, so G is for IgG. It is the guardian. It goes across placenta and it is greatest in serum, greatest in amount. Okay, IgA, it is present in airway mucosa, intestinal mucosa, and all the secretion and in the breast milk secretion. IgM is the first one to be secreted. It's massive, it's huge, and it's marker of a recent infection. IgE, it's present in emergency like your allergies, your anaphylaxis associated. It also associated with eosinophil. Okay. Now IgD. IgD, whenever you think what is the function of IgD, you're doubtful. Remember, it's present as a B cell receptor. On the B cell, IgD is present as a receptor. Clear? Now, apart from IgD as a receptor, sometimes you also have IgM as a receptor. Now, when IgM is present as a receptor on the B cell surface, that is plasma cell, it is present in a monomeric form. Only one molecule of your antibody is present. But when it is present in your serum, it is massive, it is huge, it is in pentameric form. Okay, it is pentameric, that is 5 antibodies, 5 immunoglobulins held together by the J chain. Now in your MCQs you can get this figure and they'll be like, which antibody is this? Remember, it's the IgM. And if it asked you to identify this chain, what chain is this? This is the J chain. Okay. Now IgA also exists in two forms. When it is present in the serum, it's in monomeric form. That means only one molecule of immunoglobulin is there. But when it's present in the secretions, it is present in the dimeric form. That is two molecules. You see, 1 and 2. One antibody, immunoglobulin, and second immunoglobulin. They are held together by J chain and also have this S is the secretory component. Understood? Clear? Now, let's revise everything we have gone through till now. Antibody is a Y sword. It's like a Y sword which have 4 chains. Okay? Which 4 chains? 2 identical heavy chain plus 2 identical light chain. Each antibody has the amino terminal at the upper end and carboxy terminal at the lower end. Constant region of heavy chain decides the class of antibody. Okay, 5 class: IgM, IgA, IgG, IgD, IgE. Okay, to remember the mnemonic is gamed. Clear? Next, variable region has hotspots which makes the paratope. Okay, and what does this paratope do? It handshakes the epitope. It grabs the epitope. Okay. Clear? I hope this was helpful. Like, share and subscribe for more such videos. Thank you.